cancer integrated biology. And we acknowledge the funding from NIH and NSF for this project, and also the former trainees that contributed to the completion of this project. Thank you for your attention, and I'd be happy to answer any questions.
Thank you very much, Dr. Hossein Thwana, for the wonderful presentation. So now the session is for questions, is open for questions. Any questions from the participants? Any questions you can write in the chat box, or else you can directly ask Dr. Hossein Thwana. Any questions, please? It seems no questions for you, Dr. Hossein. Thank you very much for joining us.
Well,
We are moving to the next presentation. It's from Dr. Mancini from Washington State University. Unfortunately, Dr. Mancini is not available online, but he has sent us the video of his presentation on exploiting cancer metabolism with stimuli-responsive toll-like receptor agonists. Please hang on a moment. I'll play the video from.
Hi, everybody. My name is Rock Mancini. I'm an assistant professor at Washington State University, and I'm jointly appointed there between the Department of Chemistry as well as the Jean and Linda Volin School of Chemical Engineering and Bioengineering. Today, my talk for you is titled Exploiting Cancer Metabolism with Stimuli-Responsive Toll-like Receptor Agonists. Slide to advance. So a little bit about my group at WSU. We're interested in the growing field of chemical immunology. Namely, we develop new ways to chemically manipulate the immune system. To date, we've generated a few different classes of stimuli-responsive immunostimulants. We've generated immunostimulants that respond to enzyme activity, others that are activated by ligand-receptor interactions, or avidity, and a third class that are autoregulated by cell pyrogenesis, so temperature-directed immunostimulants. Of these three classes, the most well-established by our group are enzyme-directed immunostimulants, and that's what I'm going to focus on for our talk today. So within enzyme-directed immunostimulants, you can think about the idea of targeting immunostimulants to a drug-resistant disease type. And this is a sort of well-established paradigm, particularly in the field of immuno-oncology, where immunostimulants are, immunotherapeutics are orthogonal to drug resistance or traditional mechanisms of chemoresistance specifically. So our approach to targeting the action of immunostimulants is to chemically link them to drug resistance mechanisms in cancer. So you can kind of think about how this might evolve. If you were to, say, go into the lab and design a drug, you might start with a cancer cell type that you were trying to target. You would identify some sort of druggable targets on the cancer cell itself. This might be an extracellular protein or receptor, such as HER2 in, say, breast cancer, or some sort of intracellular process, such as the topoisomerase that doxorubicin would target. You develop a go through a series of rational drug design steps to generate antibodies, peptides, proteins, or small molecules that ultimately would result in targeted cell death. However, the targeted cell death would also select for subpopulations of the cancer that have acquired drug resistance. There's several mechanisms through which acquired drug resistance occurs in cancers. A couple of them are illustrated on this slide, namely if there's a protein that is targeted by a particular cytotoxic drug or an antibody, the cancer responds by downregulating that particular target. And the mechanism of drug resistance that we exploit in my group is this idea that cancer cells have irregular metabolisms, and they also overexpress a wide range of drug efflux pumps in response to chemotherapeutic drugs. And it should be noted that this mechanism of drug resistance is actually more broad than cancer. We use a few different types of drug-resistant cancers as a test bed, but this acquired drug resistance, it reduces the efficacy of antivirals, antibiotics, and chemotherapeutics alike. So, as I mentioned, our solution to this is to use an immunotherapy with efficacy that's orthogonal to acquired drug resistance, and then target the action of that immunotherapy to drug resistance itself. So ideally, the scenario would look something like this. We have a prodrug, and for our class of immunostimulant, we've chosen the toll-like receptor agonist family of immunostimulants. These immunostimulants are caged with a directing group that can be removed by the metabolism of the drug-resistant cancer itself. This results in an immunostimulant metabolite, which is trafficked to the extracellular space through the upregulated drug efflux mechanisms in drug-resistant cancers. And once in the extracellular space, the immunostimulant can go on to activate nearby bystander immune cells from the tumor-infiltrating lymphocyte population. This conversion can occur either intracellularly or extracellularly, and we have examples of both types of TLR agonist prodrugs. Today, I'm going to talk to you about a small catalog of TLR agonist prodrugs that we've made that are converted intracellularly and then effluxed from within the drug-resistant cell to affect nearby immune cells. So let's begin with the design of a smart TLR agonist prodrug. First, what we need is an immunostimulant with an enzyme substrate attached to it. So which immunostimulant should we choose? There's a wide range of small molecule toll-like receptor agonists out there. And for our initial studies, we chose the Imidazoquinoline family of TLR agonists. There's a bit of history behind why we chose this particular class of TLR agonists. Suffice it to say that the structure-activity relationship for the imidazoquinolines is very well established. There's been a career's worth of work performed by Sunil David, who has shown unambiguously that the primary amine and immunoquinoline portion of the imidazoquinoline are critical for activity. So if you modify the primary amine really at all, you pretty much lose all agonism of toll-like receptor 7 or 8. So here's a crystal structure of an imidazoquinoline in a TLR8 homodimer complex, and you can see that the nitrogen on the imidazoquinoline is really bridging this homodimer interface, bringing it together, helping the homodimer to activate. chosen imidazoquinolines as our immunostimulant. Next, we had to decide which enzyme directing groups we wanted to choose for our TLR agonist prodrugs. And for that, we turned to the Human Protein Atlas, namely the human pathology portion of the Human Protein Atlas. So this is a catalog of thousands of patient biopsies, patient tumor biopsies that are screened for a variety of different proteins. And you can see that there's these three enzymes, an a-Mannosidase, a type of beta-galactosidase, and a beta-glucuronidase, that are widely overexpressed in a wide range of cancers. So the x-axis here, if it's too small for you to see, these are just different types of cancers. And the y-axis is the percent of patients from the protein atlas that expressed a particular enzyme. So with the enzyme in mind and the immunostimulant chosen, we could then begin to design TLR agonist prodrugs that would be activated by the corresponding enzymes that are overexpressed in these cancers. And I'm going to show you here on the slide 3 that we've designed. We call them RSQ-Flu, RSQ-Man, and RSQ-Gal. The RSQ stands for resiquimod, which would be the parent immunostimulant here. Depending on what the R groups are, you could either have resiquimod, or if R1 and R2 are equal to H, then you would have imiquimod as a prodrug. And we've made the imiquimod, resiquimod, and gardiquimod versions of each one of these. And I also want to point out, in the realm of enzyme-directed immunostimulants, our current work in progress is an immunostimulant that's directed by beta-lactamase. Regardless, each of these can be converted via enzyme activity to a highly immunogenic TLR7/8 agonist of either imiquimod or resiquimod. So I understand this is a pharmacy conference, so I'll go a little light on the synthesis here. Suffice it to say, in my lab, it typically takes a first or second year grad student just under a year or so to work out a new synthesis of an enzyme-directed TLR agonist prodrug. And once the steps are kind of sorted out, then these can be produced on the order of weeks. So you can see that the syntheses, they all begin from the parent carbohydrate, so the enzyme substrate itself. The substrate is protected, and the anomeric position is brominated with HBr and glacial acetic acid. Then a self-immolative spacer is installed, so orthonitrophenylaldehyde. The spacer itself is reduced with sodium borohydride to the corresponding alcohol, and from the corresponding alcohol, we can make an activated carbonate with paranitrophenylchloroformate, and then these activated carbonates are susceptible to nucleophilic attack, and this is useful because if you look into the literature for toll-like receptor agonists, you'll find that there's a wide range of nucleophilic moieties that are critical for the activity of TLR agonists. So while we use the imidazoquinoline class as sort of a model system, we really envision that these enzyme directing groups could be used on any number of TLR agonists that are out there. Regardless, the next step is to couple to the nucleophilic nitrogen on the imidazoquinoline. This is typically accomplished in our lab under microwave conditions. And then following deprotection of the carbohydrate hydroxyl groups, we can arrive at the corresponding enzyme-directed TLR agonist prodrug. To date, we've made versions of these enzyme-directed TLR agonists that are activated by Mannosidase, galactosidase, glucuronidase, and most recently beta-lactamase, which in the interest of time, we won't walk through the whole synthesis here. Suffice it to say, we can obtain a beta-lactamase-directed immunostimulant in acceptable yield. So with these enzyme-directed TLR agonist prodrugs in hand, we next wanted to see, well, does exogenous enzyme actually cause conversion from prodrug to immunostimulant? And ultimately, do cancer cells or bacteria metabolize these molecules to immunostimulants? For that, we turned to a raw blue cell assay. For those that are unfamiliar with raw blue cells, this is a cell line that is transfected to express all of the toll-like receptors with the exception of TLR5, and activation of NF-κB or AP1 is coupled to a secreted alkaline phosphatase output in these cells. So effectively, activation through the toll-like receptor pathway results in production of alkaline phosphatase, which can be measured via colorimetric assay. So we took each of our prodrugs and fed them to the corresponding enzyme, and then and first monitored conversion by LCMS. So that's the red circles here, as compared to just incubation without corresponding enzyme. So the background rate of hydrolysis is rather slow. Over 48 hours, there's minimal hydrolysis that occurs, 3 to 5%. However, in the presence of corresponding enzyme, there is full conversion in every case to the corresponding immunostimulant. The immunostimulant itself is bioavailable and capable of activating raw blue cells. So the colorimetric assay that we just discussed is mapped on the y-axis here, and this is the solid colored bars over time, as compared to the lightly colored bars where we do not add exogenous enzyme. You can see there's not really very much activation at all. So this is good. This proves that the enzyme directing group effectively masks the activity of the immunostimulant, and that when the enzyme directing group is removed by enzyme, the corresponding liberated immunostimulant activates immune cells. Okay, so we next asked, could we actually use MDR multi-drug resistant cell metabolism to cause this conversion and ultimately provoke an immune response? So for that, we used AT3B-1 rat prostate cancer cells. This cell line is kind of neat in that you actually grow them in 1 micromolar doxorubicin chemotherapeutic. And the cells here were fed our prodrug. Here, the carbohydrate we used was mannose, so this is conversion by a-Mannosidase. And then following conversion to active immunostimulants in the cell media, we transfer the supernatant to our raw blue assay to measure the effective immunogenicity that's generated by this conversion. So you can see in the graph here, if we take our enzyme-directed immunostimulant and just feed it exogenous enzyme, there's an immune response that rapidly grows in over time. Instead, we take our enzyme-directed immunostimulant and feed it to a chemo-resistant AT3B-1 rat prostate cancer cells. You can see that there's also an immune response that grows in over time. And both of these immune responses are sensitive to the addition of swancinine, which is an a-Mannosidase inhibitor. And so this proves that we're going through a-Mannosidase conversion in AT3B-1 cells. And as a sort of initial test of specificity, we looked at conversion of our a-Mannosidase prodrug in raw blue cells themselves, and we see minimal conversion or minimal immunogenicity over 48 hours. Okay, so this prodrug is being converted by the cancer cells, but how is it actually entering following conversion. And we don't have time to go through all of the experiments we've done so far. We're getting ready to publish a paper on this, so be on the lookout if you're interested. But suffice it to say, we think that the trafficking to the extracellular space is at least occurring through P-glycoprotein-mediated drug efflux, in addition to a few other pathways. We've looked at this in a variety of ways, the most telling of which, though, is competition assays with rhodamine 1,2,3, which is a known fluorescent P-glycoprotein substrate. We've also looked at this sort of mechanism using a variety of different enzyme substrates. So we've looked at the glucuronidase pathway, the Mannosidase pathway, the galactosidase pathway, and crossed those pathways with a variety of different cancers, namely B16 melanoma, TRAMC2 prostate cancer, or 4T1 breast cancer. We can see there's an appreciable immune response that's generated in each case, with B16 cells really having the highest activity out of the three cancer types for this sort of scenario that I've illustrated here. We tried to look into this a little bit further to see, well, okay, is it that the efflux pathway is upregulated, or that the actual enzymatic conversion is upregulated in B16 cells? The best we can tell, we think that it's efflux that's the rate-limiting step in our scenario of feeding enzyme-directed immunostimulants to cancer cells. So we've developed a working model that looks like this. There's uptake by cancer cells through a mechanism that we have not conclusively proven yet. The immunostimulant is converted through cancer cell metabolism. The pro-immunostimulant is converted through cancer cell metabolism to active immunostimulant. The immunostimulant is then effluxed, and we think this is the possible rate-limiting step here. Efflux to the extracellular space, where it can go on. We generate multi-drug resistant cancer cells that have enhanced mechanisms of drug efflux. We should also enhance this mechanism of bystander-assisted immunotherapy that we have as our working model for how our enzyme-directed toll-like receptor agonists function. So this was essentially what one of my graduate students, Anune Kulakori, did during the COVID-19 shutdown. He was listed as essential personnel, and he just came in religiously, slowly culturing our cancer cell lines to become doxorubicin resistant. By nature of acquiring this resistance, they begin to overexpress these efflux proteins. So here's just how long it took him to culture each of these cells: B16 cells, TRAMC2 cells, and 4T1 cells. And running a Western blot for P-glycoprotein, we can see that indeed, relative to the parent cell lines, the multi-drug resistant cell lines that we've generated do indeed express more P-glycoprotein than the parent non-MDR cell lines. P-glycoprotein is there in the parent cell lines. You can see that if you really overexpose the blot. However, it's clearly upregulated in the multi-drug resistant cell lines. Okay, so that sort of leads us to where we are right now, as we currently have these multi-drug resistant cell lines in hand, and we are currently repeating this same sort of study where we feed our enzyme-directed prodrugs to this multi-drug resistant cell panel to observe which enzyme substrate is converted the most efficiently, as well as which immunostimulant is effluxed most efficiently. For the results of that, the best I can say is that they're coming soon, and be on the lookout for it in summer of 2021. And so hopefully I've shown you today that synthetic organic chemistry presents a powerful tool to manipulate the immune system. And I've shown you one category of 3 in which we're doing this in our lab. With that, I'd like to thank my group members. Their names were highlighted on the different slides throughout the talk. I'd like to thank my collaborators, as well as WSU, the AAI, NIH, and MJ Murdoch Foundation for funding. With that, I would normally pause to take questions, but since this talk is pre-recorded, in case there are technical difficulties, please feel free to send me an email if you have any questions. Thank you.
Okay, that's a talk from Dr. Mancini. Unfortunately, he's not available online. If any questions, I'll take the questions and I'll just forward the questions to Dr. Mancini. Does anyone have questions? Anyone has questions? You can write down in the chat box. I can take the questions and I'll just forward to Dr. Mancini. Okay, if no questions, then I'd like to invite the next presenter. It's Dr. William Gregory from University of Wisconsin, Milwaukee, USA. Dr. William Gregory will be speaking on the core relaxation frequency as a prognostic parameter for breast cancer. Dr. Gregory, over to you.
Can you hear me now?
Yes, I do.
Okay. I need to,
Share your screen.
yeah, let me share the screen there. Okay. I don't see the share the screen. Wait a minute. Share screen here.
You need help from our side?
Let me see.
Yes, now I can see your screen. You can just open your presentation.
Are we there?
Yes. Can you just open your presentation?
I did.
But we can see the folder view. I can see a lot of PPTs over there.
You do not see the, that's strange because it is.
Okay, you can just double click on that.
Says your screen is paused.
First, open your presentation.
Yeah, I've got it open.
Then you come back to Zoom and you can go for a share screen option.
Oh, I see. I opened the presentation.
Then you come to share Zoom window again.
Can you see it now?
No, I can't see it.
It's strange.
Can you just close it down and repeat the process again?
Okay.
So just open your PowerPoint presentation.
Okay. I got it open, and I have it open.
You already opened it.
It's open. It's open now.
Okay, then you come back to a Zoom window. Click on share screen option. You can see the open windows. There you can find a PowerPoint presentation.
You
Okay,
see it
now I
now?
can see that, yes.
Okay.
Okay, you're good. You can go ahead.
Yeah, okay, now if I get it to... Okay, there we go. All right, well, thank you for the invitation today to speak. I'm sorry that this has gotten a little bit confused. This is a report of a follow-up of a study we did in Milwaukee at the what was then the Aurora hospital system, and has now become Advocate Aurora. The work reports a follow-up to what's called an EPET-1, and that means an electrical property evaluation technology, where we studied the electrical properties of tissue that had been excised during breast cancer surgery. And the patients were consented and asked to participate with follow-up medical visits. It was then, which would then be recorded in the Aurora Cancer Registry. An average of about 8 years of follow-up data were acquired over a 10-year span. Reference 1 is the result of that study. The follow-up is a recently published brief report, and it is there's a link, and by the way, the link is free. So, as I said in Mad Magazine, for free take for buy waste time. So in this presentation, I'm going to review both of those studies, because a lot of people will probably not be completely familiar with them, and then give you my take on a hypothesis to explain them. Okay, so the first question I have is, what is unique about tissue electrical properties as they're compared? Are they comparable to inanimate materials? And of course, the answer is no. They're not, and if you took a whole bunch of chemicals, put them in a pot, stuck some electrodes in them, and measured the electrical properties, they would not have any really relation to this, what you see with the tissues. Of course, the reason for that is, simple answer to this is the cell that is the difference. And we know what that is causing that difference. It's the cell membrane. This is a picture of the cell membrane, rather stylized, but showing a lot of stuff goes on in the cell membrane. Most importantly, the membrane itself is a double layer of lipids, so arranged that the interior side, that lipid is what they call water. It doesn't like to have water, but the two sides it do, and so they connect. And there's very little electrical conduction that goes on in there. The resistance is extremely high. However, the charges on that side, a positive charge would produce negative charges on the inside. And given that there's very little resistance here, if the resistance was a very high resistance, that would then make a rather good electrical capacitor. And so what do you do to make such use such a measurement? What does it tell you about the cell? Well, first of all, what to do is you measure the voltage drop across the tissue divided by the current, and it's called the impedance. And you can see that if you have current coming in through the interstitial fluid or matrix, when it gets to the cell, it's either going to have to go through the membrane, through the capacitor, or going to have to go around. Okay, so you need to decide how to do that electrically. And this is a fairly simple circuit. Others have been proposed, but this one works well enough for this presentation. The tissue comes in